Okay, so we've discussed adsorption, we've discussed penetration and uncoating. Now for the third step, we have viral genome, the DNA or the RNAs in the cell. Now it's time to start making copies. Copies of everything. Copies of the genome, copies of the capsomeres, whether it's going to be for icosahedral capsid or for the helical capsid. Couple of rules of thumb here when it comes to viruses. If it's a DNA genome virus, that DNA is going to be trafficked, it's going to be transported into the nucleus. Because once there, nucleotides are nucleotides. Our cells cannot tell the difference inside the nucleus of what is human and what is viral DNA. So your own enzymes will start making copies of the viral DNA. DNA polymerases make more and more DNA virus genomes. The RNA polymerases will read those viral genomes and make messenger RNA. The tRNA, the ribosomal RNA, genes for that's not there because it's going to be using host. Now, if it's an RNA virus, it's going to stay in the cytoplasm. Okay? RNA has no place in the nucleus. As soon as messenger RNA is made, out the nuclear pore it goes. Sometimes it's not even finished being made as it's moved out. So, turns out the RNA viruses are going to have a few extra steps. Okay, think back to a previous lecture on viruses. There's going to be RNA-dependent DNA polymerase. RNA-dependent, meaning it reads RNA. DNA polymerase means it makes DNA strands. Reads the RNA, makes DNA. Once the DNA is made, that's transported. So what steps are necessary? Well, it's going to depend upon which virus you're talking about. And we'll get more into the in-depths of that when we're talking diseases, because it turns out, comes the RNA viruses, these different new steps that have to take place are great targets for antiviral compounds. Not antibiotics. Antibiotics are for bacterial infections. Antivirals are for against viruses. So, once you have capsomeres made and they start to self-assemble, they can only go so far with their self-assembly until you have a genome that's going to fit up in there for it to complete. And when it comes to viral assembly, it all just happens automatically. Turns out capsomeres have an affinity for each other. Once they start to form that triangle shape for the icosahedron, turns out the triangles now have a high affinity for other triangle assemblies of capsomeres, and it just keeps going and going. The helical, well, turns out the capsomeres have an affinity for each other, but they have to have a piece of DNA or RNA to assemble around, or it just keeps falling apart. Release is going to be important. Release of the viruses, that when it comes to enveloped viruses, turns out these have more steps they have to go through. Viral proteins that get inserted into the plasma membrane. The icosahedron or the helical nucleocapsid start to have an affinity for these viral proteins. Some of these proteins are the spikes in the plasma membrane of the host cell. As more and more of the nucleocapsid interacts with those spikes, those viral proteins, it starts to wrap around. As it begins to wrap around, as you see, it distorts and it pushes out the plasma membrane. And it's going to get to a certain point that the plasma membrane of the host cell stretches, and now it's either going to get tear or it's going to release and reseal instantaneous. Far side is an artist rendition above my head. Actual microscopic pictures showing you this process.